Reproductive handicap when you consider sexual reproduction because only half of the members of the population are able to reproduce to produce offspring. Males contribute genetic material, but they're not actually producing. So over time, an asexual population can produce many more offspring. Well, what's the advantage? So sex must enhance reproductive success or survival, or it would rapidly disappear. So in sexual reproduction, it requires the fusion of gametes and uses the process of meiosis to create haploid gametes with half the chromosomes. Offspring produced are different from the parents because of new genetic combinations resulting from fertilization followed by meiosis. So while asexual reproduction will produce many more offspring, it's only going to be advantageous in a stable or unchanging environment. The variation that sexual reproduction produces is a benefit that makes it evolutionarily successful. It's a shuffling of genes that allows the populations to rid themselves of harmful genes and to accumulate beneficial mutations in a much more reliable way. Through reproduction, you produce clones, a group of genetically identical individuals. Members of a clone may be genetically different, though, due to mutations. There are many different types of asexual reproduction. It doesn't involve the fusion of gametes, but you have hydra, for example, budding. You have plants going through vegetative propagation. You have organisms like the whip-tailed lizards or bees that use parthenogenesis as ways where you can develop asexually, and so it gets really complicated. Parthenogenesis is an interesting one because an egg is developing there without being fertilized. It occurs in bees and wasps and ants and lots of things, with the offspring developing into adults that produce egg or sperm without meiosis. For example, with honeybees, male drone honeybees are fertile haploid adults that arrive by parthenogenesis, while female honeybees, including both the sterile workers and the fertile queens, are diploid adults that develop from fertilized eggs. We have observed this, like we said, in other species like Komodo dragons and whip-tailed lizards and a few other pieces. So it is definitely something that we are still trying to learn more about. Have the potential to reproduce quickly in favorable environments, and while most prokaryotes have generation times between 1 and 3 hours, some species can produce a new generation in 20 minutes under optimal conditions. A single cell in favorable conditions produces a large colony very quickly, asexually via binary fission, with the synthesis of DNA almost occurring continuously. Prokaryotic reproduction is limited because cells eventually exhaust their nutrient supply, accumulate metabolic waste, face competition from other microbes, or even consumed by other organisms. But it draws attention because of three key features of their biology. They are small, they reproduce by binary fission, and they have short generation times. Their ability to withstand harsh environments is a major factor of why we're looking at these. And mutation is their major source of variation. So with that very short generation time, they're able to adapt very, very quickly and are actually highly evolved. It's actually have extensive genetic variation, but it results from rapid reproduction and mutation. So when they reproduce by binary fission, some of the offspring differ slightly. These spontaneous mutations only occur every so often. The exact number is kind of hard to determine, but the genetic diversity can lead to rapid evolution from mutation. Additional diversity arises from genetic recombination, which is going to be the combining of DNA in different individuals into a single genome. Now, this could be something with the actual chromosome, that circular chromosome that the bacteria have, or it could be small segments of DNA called plasmids that can be exchanged, which you can see in the picture on the right. But overall, what you're looking at here is the movement of genes from one organism to another, or horizontal gene transfer. And bacterial recombination occurs through three processes that we'll look at next. Transformation is the alteration of a bacterial cell's genotype and possibly phenotype by the uptake of foreign DNA from the surrounding environment. The DNA may come from dead, broken open pathogenic cells, but you have the foreign allele replacing the native allele in the bacterial chromosome by recombination of homologous DNA segments. The resulting cell is now recombinant with DNA derived from two different cells. Researchers have learned overall that bacterial species have surface proteins that are specialized for the uptake of DNA, and they recognize and transport DNA from closely related bacterial species into the cell to allow that incorporation. The second major source is transduction. Transduction is when a phage, a virus that infects bacteria, carries prokaryotic genes from one host cell to another due to aberrations in the phage reproductive cycle. The virus that carries prokaryotic DNA may not be able to replicate because it lacks some or all of its own genetic material. However, it can attach to another prokaryotic cell, inject the DNA that is acquired, and then reproduce that way. But because new DNA is being incorporated, a recombinant is still being formed. Horizontal gene transfer is going to be conjugation, where you have the transfer of genetic material between two bacterial cells, usually of the same species, that are temporarily joined. They're going to be joined by these hollow pili, these mating bridges. The ability to form and donate DNA results from an F factor, F for fertility, and it's a section of the bacterial chromosome or as a plasmid. It's only about 25 genes, but without it you can't produce this structure. A cell lacking the F factor, designated an F minus, functions as a DNA recipient. Transfer of that entire plasmid converts an F minus to an F plus and can then be integrated into the bacterial chromosome. Overall, when chromosomal DNA from these cells enter, homologous regions are aligned, allowing segments to be exchanged, resulting in, again, the production of a recombinant bacterium with genes derived from two different cells. Cycle is the generation-to-generation sequence of stages in the reproductive history of an organism. We understand the human life cycle most of all, with meiosis producing haploid gametes that come together to form a diploid zygote at fertilization, which will then divide through mitosis and development to produce the organism and to maintain that cycle. But it's all about DNA exchange and crossover and variation and production within those structures. So when you look at the chromatograph or the picture of those chromosomes that you see on the right, we can see that the picture that you think of is going to be those duplicated sister chromatids. At metaphase, they're pulled out manually and rearranged in these slides for this structure, so we can see what's going on there. Life cycle is one that we understand, but it's not the only life cycle that exists. We need to understand that humans, we have XX is female, XY is male, the two different sex-determining chromosomes occurring there, but it's really only a single gene, the SRY gene, that makes the difference. So if you were to remove the SRY gene from a Y chromosome, you would still produce a female offspring. Other organisms use different combinations of these things. With chickens, for example, being ZZZW, the male having two copies and the female having only one copy. And then you have the bees that are producing, and fruit flies and wasps that are producing parthenogenetically, and a variety of other concerns. So the XY relationship, XX relationship that we're aware of in humans is common in many mammals, but it's far from the only one that exists. Reduces the number of chromosome sets from diploid to haploid. It is a reductive process. You're going to go from 2N to N. Many steps here resemble mitosis because it evolved from mitosis, but both processes are preceded by the duplication of chromosomes. In meiosis, there are two consecutive cell divisions resulting in 4 daughter cells. The first, meiosis 1, separates homologous chromosomes. The second, meiosis 2, separates sister chromatids, just like mitosis. The 4 daughter cells at the end have only half as many chromosomes as the original cell because there is no duplication phase between meiosis 1 and meiosis 2. There is no interphase between those structures. Overall, 2 genetically identical sister chromatids make up 1 duplicated chromosome. The sister chromatids are closely associated all along their length, and then those are eventually going to be split through this process. Homologous chromosomes appear to be alike, but they have different versions of genes called alleles at corresponding loci or locations on the chromosome. For all meiosis 1, you can see the structures and the processes that are happening here. Prophase 1 is when the chromosomes and the tetrad align together. That overlap process is called synapsis. Where they overlap is the chiasmata, and then that's going to allow crossing over to occur, which we'll look at in a moment. Those chromosomes will then align in metaphase 1 at the metaphase plate, where they will be split during anaphase, so the homologous chromosome pairs separate and move to opposite poles, and then telophase 1 and cytokinesis will occur with these chromosomes. At the end of this process, they already are down to a haploid structure, but the chromosomes are still duplicated. So we went from having 6 chromosomes to having 3 chromosomes. And now we move into meiosis 2. In meiosis 2, this is a lot like mitosis, except that it's happening twice with haploid cells. So the sister chromatids are going to separate, resulting in 4 haploid daughter cells that we call gametes, each with a single unduplicated chromosome. If you apply this to actual cells, spermatogenesis is going to be the production of sperm, and it's going to be very, very close to that full process. The primordial germ cells are going to be producing sperm all throughout the lifespan of the human in these seminiferous structures. So you have the spermatogonium, which is going to produce the primary spermatocyte, which is then going to produce all of the rest of those levels. So with spermatogenesis, the haploid structures that are produced, one spermatogonial stem cell will produce, in the end, 4 sperm cells that are haploid and can be used for sexual reproduction. Oogenesis, or production of the egg, is a little different. You produce 1 haploid egg and 3 polar bodies. Women are born with every egg they will ever have, and they're basically frozen in prophase 1 until puberty. 1 to 2 are going to be released every month after puberty, and they're going to be frozen at metaphase 2 until fertilization. So overall, oogenesis is going to produce those structures, and that's going to be part of that reproductive cycle. We looked at the menstrual cycle when we looked at cell communication, and this is going to be part of that process as well. So overall, spermatogenesis differs from oogenesis in 3 major ways. In spermatogenesis, all 4 products develop into mature gametes. In oogenesis, the cytokinesis during meiosis is unequal, with almost all of the cytoplasm segregating into a single daughter cell. This large cell becomes the egg. The other products of meiosis, smaller cells, are called polar bodies and will be degenerated. Spermatogenesis occurs throughout adolescence and into adulthood, and then during oogenesis in human females, mitotic divisions are complete before birth, and the production of mature gametes ceases at menopause. Spermatogenesis produces mature sperm from precursor cells in a continuous sequence, whereas oogenesis has long interruptions. So overall, all of the cytoplasm and all of the cellular machinery end up in that one egg, and that's going to give it a great deal of advantage for early survival. And the remaining bits, the polar bodies, are essentially just the unneeded genetic material. Within meiosis, there are a couple of sources of variation that exist that make it so advantageous. The first part happens during prophase 1. So when those duplicated chromosomes come together in prophase 1, they overlap really tightly in a process called the synaptonemal complex. Where they actually connect, the chiasmata, those are going to be crossover points, and the DNA can break and rearrange within that structure. It's called crossing over, and it's a major source of variation in sexual reproduction. So as a result of this process, individual chromosomes can carry genes derived from 2 different parents. So it's going to produce new combinations of genes and allows for a great deal of variation. In addition to that, independent assortment basically says that there is genetic variation because of how the chromosomes line up at the metaphase plate. There is a 50-50 chance that a particular daughter cell from meiosis 1 will get a maternal chromosome, and a 50-50 chance that it will get a paternal chromosome. Each of those chromosomes exist independently of each other and how they align, so independent assortment in metaphase 1 produces 2 to the 23rd different combinations in each person's gametes for humans, because we have 23 pairs. That means there's about 8.38 million combinations just from independent assortment. If you then combine that with fertilization, because it's random which cell you get from fertilization, that's 2 to the 23rd times 2 to the 23rd, that means that there's about 70 trillion possible combinations between any 2 people. If you then apply crossing over to that in addition, the difference is functionally infinite, and the major source of sexual selection going on, the sexual variation, is happening because of these processes. So mitosis versus meiosis is very important to understand. During meiosis, the sister chromatids are attached along their lengths. In mitosis, the enzymes remove those connections to allow the sister chromatids to move to opposite poles. In meiosis, instead, the sister chromatid cohesion is released in 2 steps in those different processes, anaphase 1 and anaphase 2. Mitosis is a non-reductive process that produces 2 diploid cells, and meiosis is a reductive process that can produce up to 4 haploid cells. So meiosis is reductional, mitosis is not. The equations and structures and process, they seem very, very similar because they evolved from the same sources, and you sort of get the idea, I hope. So obviously this is occurring for humans, where we have an N of 23, so our 2N would be 46. This picture here is just showing a 2N of 6, but it should show you the rough idea.